Hi there and welcome to Mr. Fisher's Biology Podcasts. This podcast is on protein synthesis. So protein synthesis is the production of proteins, and it involves two phases. In protein synthesis we find transcription and translation. And before this takes place, mRNA needs to be processed. So we need to do a bunch of things before it leaves the nucleus in order for the synthesis of proteins. So the pathway to making a protein can simply be broken down into three processes. So we have DNA to mRNA, and this process here we call transcription. And when we convert mRNA into a protein, this is translation. This takes place in the nucleus of eukaryotic cells. And this takes place at ribosomes within the cytoplasm of cells. So let's talk a little bit about RNA first before we get into the synthesis of proteins. There's a couple of notable differences from RNA and DNA. First of all, DNA has a deoxyribose sugar and RNA is a ribose sugar. RNA contains the base uracil, where DNA has the base thymine. And RNA is a single-stranded molecule and DNA is double-stranded. There's two different types of RNA that we're going to talk about in this course. The first of those types is messenger RNA, or mRNA, and mRNA carries genetic information to the ribosomes. So think of mRNA as a photocopy of DNA. Now, transfer RNA is a different type of RNA, and transfer RNA is specific in its job in that it will take amino acids to the ribosomes where proteins are synthesized. So transfer RNA will bring amino acids to ribosomes, and those amino acids will then join to form polypeptides or proteins. So when we're making a protein, we're going to talk about genes first. So proteins are made of amino acids. And amino acids, remember, are linked together by peptide bonds. If we look in the world, there's 20 different amino acids. When we combine amino acids into chains, we call that a polypeptide. It's not a protein yet. It's got to undergo changes to become a protein. Okay, and we call those changes folding. Okay, so in order for a polypeptide to become a protein, proteins need to undergo some modification, or polypeptides need to undergo modifications. Now, within the DNA code, we find a specific sequence of nucleotides that code for a protein. And that code for a specific protein we call a gene. Now, as I said before, there's two parts to protein synthesis. So transcription is making an RNA model or a photocopy complementary to DNA, and translation is taking that mRNA and converting it into a sequence of amino acids. And remember, those sequence of amino acids will eventually become a polypeptide, which will then eventually make a protein. If we look at the genetic code, on the DNA strand, we find a triplet code. That means that every 3 bases on a DNA strand code for one amino acid. And every 3 letter units on an mRNA we call a codon. So DNA, it's a triplet, and mRNA we call a codon. Most amino acids have more than one codon. There are 20 amino acids. There's 64 different triplets that we can make out of those 3 letters, and the code is nearly universal among living things. So for example, if I look at a DNA triplet, let's say I have cytosine, guanine, and thymine, and that's on my DNA strand. This is going to code for an mRNA codon, and that codon is going to be complementary, keeping in mind that within mRNA we lose are thymines for uracils. So a cytosine will become a guanine will become a cytosine, and a thymine, because it's in DNA, will code for an adenine as a codon. So this is the triplet, and this is the codon. Okay, we'll talk a little bit more about that in a little bit. But we first come to this picture right here. And this picture is showing us a mRNA code. It shows us how we can read the codon for a specific amino acid. So if we take cytosine, guanine, and adenine, which was our example on the last slide, let's find what amino acid that codes for. So first of all, I look in the first base, and I'm going to cytosine. That's my first base. I then move along the second base and look for guanine, which is right here. So I'm looking in this box. And now I look for adenine. Well, adenine is the third base, and it's right here. So this is going to be arginine, the amino acid arginine. Now also notice I could have made arginine with any nucleotide in the third part of that codon. And this is what we mean by there are redundancies in the code. So there are codes, there are numerous codes to code for the same amino acid. And in this way, when we talk about mutations, we'll find out how useful it is to have those redundancies built in. So to go over this really quickly again, we have our DNA double helix strand. When that opens up, we will get transcription. So we will get DNA to mRNA. Now the DNA triplet, which we can identify there's 3 triplets on this DNA strand, code for 3 different codons, which we call codon 1, codon 2, and codon 3. I'd like you to stop the podcast and see if you can use your genetic code wheels to identify what these 3 amino acids are. So if you would have done this, you would find that CGC codes for arginine, and I'm just going to do short form, ARG. UAG, that codes for something called a stop, which is a special type of codon that tells ribosomes to stop producing. And ACC codes for an amino acid known as threonine. Hopefully you got that. A stop codon would never exist in between other codons like this. We tend to find stop codons at the end, but this is a good example of transcription to translation, and this is something you need to know how to do. So when we look at transcription, DNA unwinds and unzips, serves as a template for mRNA. We have an enzyme called RNA polymerase. Do not mistake it for DNA polymerase. And it joins RNA nucleotides so that the codons are complementary to the triplet code in DNA. The transfer of information in the nucleus from DNA to RNA, that's transcription. Only one DNA strand serves as a template, and it starts at a region on a DNA strand which we call a TATA box. And all a TATA box is a sequence of nucleotides that form a TATA sequence. And that tells your enzymes that this is the start of a gene. It ends at a terminator, which is a stop codon. And in a lot of cases, terminators are repetitive sequences of similar nucleotides. And in that case, the enzyme would know it's time to stop. When we're completed, we have what's called a pre-RNA molecule, and it's ready to be processed. Now, RNA polymerase, remember, is the enzyme that separates the strands, and it links RNA nucleotides together on the from the DNA template. Now, when we process pre-RNA, This also occurs in the nucleus. We have segments called introns and exons. It's important to note that exons are what code for proteins, and introns do nothing. So introns are spliced out, and they're spliced out by a special enzyme called the spliceosome enzyme, and the exons rejoin. What we have is a mature RNA strand. So here is RNA processing. So notice we have a pre-RNA molecule, we have exons, and we have introns. The spliceosome enzymes will remove those introns, and they'll take them out of the strand, and we're left with a mature RNA molecule. Now, messenger RNA, or mRNA, is what will carry that information for a protein. It's made up of 500 to 1,000 nucleotides. It contains a sequence of 3 bases, which we call a codon. On messenger RNA, the codon AUG codes for methionine, and it's also the start codon, so the ribosome knows when to produce or start producing a polypeptide chain. We have talked about these before. We have 3 different stop codons which tell the ribosome to stop producing a protein. So here we have our mRNA going to different codons. Now notice that and use your code wheels to just verify that each one of these codons does in fact code for those, and this creates what's called the primary structure of a protein. This is what we call a polypeptide. A polypeptide is really just a sequence of peptide bonded amino acids that will eventually, through processing, become a protein in our body. Okay, now, in order for this to happen, we need another type of RNA, which we've already talked about, and that's transfer RNA. It's made of 75 to 80 nucleotides. It picks up amino acids. It transports amino acids to the mRNA. Now, tRNAs have an anticodon that's complementary to the mRNA codon. And this is where tRNAs will recognize the appropriate codon. Now, in this picture right here, this is where an amino acid will attach, called the amino acid attachment site. And at the bottom of our tRNA, we find an anticodon. Now, the mRNA strand that this would bond to would be AUG. And in that case, those letters are complementary to each other. We're still in RNA, remember, because we're both RNA strands. We don't have thymines, we have uracils, and AUG codes for methionine. So this, in fact, would be the tRNA for methionine. Now, ribosomes. Ribosomes are very important. They're made up of a large and small subunit, and they have 2 sites called the P and the A site. And so this is what a ribosome looks like. It's composed of a large and small subunit. This right here, the mRNA strand, feeds in to the P and the A site, kind of like a computer would read a data card. And this is where the start of protein synthesis takes place. And that brings us to translation. Translation is the synthesis of proteins in the cytoplasm. It involves mRNA codons, it involves a tRNA anticodon, it involves the ribosomes, and it also involves amino acids. There are 3 steps to translation. The first step is the initiation, which starts with the start codon. Then we have the elongation, and during elongation, amino acids are linked together. And remember, they're linked together by these peptide bonds. And then we have termination, and termination happens when a ribosome recognizes a stop codon. So let's go over the translation and how it works in a ribosome. So starting with our tRNAs, and remember that tRNAs kind of look like this. These tRNAs will come in on the P site. Remember, they recognized by a particular anticodon, and in this case it would be UAC. Another tRNA, which are free swimming in the cytoplasm, would again come into the next position. Both of those are carrying amino acids, and those amino acids would bond together with a peptide bond. Now, the first the first tRNA would leave. This one would move over. And we would get a new tRNA coming in, carrying a new amino acid, and again another peptide bond would form. And this would happen until we reach a stop. The end product is a polypeptide chain, which is the primary structure of a protein, which is a sequence of amino acids bonded together by peptide bonds. And that concludes our podcast on protein synthesis, and I hope that was helpful.